everyone. No one's in the classroom yet. This is my cat Minerva. She's helping out today. She just wants to get in my way and walk across the keyboard. So if strange things happen, that could be why. All right, now you're just going to sit here and watch me wait for people to come in. We will start. It's 1 o'clock now. I'll give people a couple minutes to get dialed in, figure out how to get in here. Cat, get your butt out of my face. And then we will start our review. So exam 1 opens, exam 2 opens Sunday at midnight, so Saturday at 12:01 in the morning, and will be available until midnight, 11:59 p.m. on Tuesday. Same format as the last exam, same rules. Make sure that I can see your face on your Respondus monitor or honor lock. If I don't see your face, or if I see your ceiling like this in your forehead, automatic 10-point deduction from your grade, because I can assume that you are looking elsewhere, like at your notes or book or something. And so make sure I can see your face the whole time. Nobody else is in the room. So you have adequate lighting. What are some of your answers? Yep, just answer. I'll check the videos later. I am so impressed. Out of all the 101 sections, we did not have a single red flag in our class on exam 1. And you were the only 101 exam section that had no red flags. Those red flags come up on our recordings, our video recordings, telling us what minute we had a red flag and what the violation was. So it could be you get up and leave the room. Again, 10-point deduction. So make sure you take care of all your needs, bathroom, water, whatever else you're going to need during the exam beforehand, so that you don't have to get up and leave. 10-point deduction. Make sure your cell phone's not around. One thing another instructor had was somebody looking down periodically, but their light from their cell phone was shining up on their face and she could see that. So don't want to see any of that. But I think you guys are good. You did well on the first one, so I am not too worried. I think everybody has a better idea how to study. You're all learning and trying, experimenting with new study strategies. So I think that is that's helpful. That our discussion, our metacognition questions, that is what they are there for. Hi, Sabrina.
Hi.
So you're the only one here so far, so I've just been here talking to myself. This is being recorded, so I'll post it later. But if at any time you have any questions, I'm just going to go through the learning objectives. So if you have any questions at any time, just unmute yourself and jump in and let me know. All right, and I'll keep an eye on as other people jump in and let them know to do the same. So otherwise, I'll just go through pretty briefly. So it's Unit 2 covers chapters 2, 3, 4, and 5. So we're looking really at building a cell from atoms, from our building blocks. And so I will go ahead and share our learning objectives. And we will just go right down the list on these. And I put some notes in here as little reminders, but these are at the top of every chapter heading in D2L. So we broke down into units and then chapters, and at the top of each chapter we have these, which should be basically your study guide. So if we start with basic chemistry, we really don't go into a lot of detail, but if it's the first time you're getting some of this, it can be hard to remember. But when we're looking at the periodic table of elements, this is what I like to say is a way to remember the 6 elements that make up about 95 to 96% of most living organisms. So these are the elements that make up most of our body, most of the dry weight, meaning if I removed all the water, meaning I say, oh, we're about 70% water. But if I removed all the water, what I would have left would be those proteins, lipid, carbs, nucleic acids, the thing we learn about in the next chapter. But the elements that make that up are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And I write them in this order because I like to say we are made of schnapps. So if you pronounced that phonetically, it would be schnapps. So we're made of schnapps. There are 6 elements. And then you should be able to describe an atom, and that's the Bohr model of the atom. Gives us a nucleus, that central portion where the protons with a positive charge and the neutrons with no charge are housed. So nucleus just means the core or the middle, and we just have to make sure we pay attention as we get going, because when we talk about cells, they have a nucleus, but that's something different than an atom's nucleus. So our protons and neutrons have 1 atomic mass unit each. That's just a random number. These are so small. They are smaller than really the ability to measure these. When we discovered those, when Niels Bohr started naming these things. So they're designated as 1 atomic mass unit, and our atomic mass or weight, there's a little slight variation in the use of these, but it's the number of protons plus the number of neutrons. And then surrounding that nucleus in orbitals, circling kind of like the planets circle the sun. So the sun would be the nucleus and the planets are the electrons. Are our electrons with a negative charge. Our elements typically have no charge unless they are ions, which that's a special case, but normally they have a neutral charge, meaning the number of protons and electrons has to be the same. And the number of protons gives us our atomic number. So the mass we know is going to be the protons plus neutrons. That's always going to be the bigger number. The atomic number will be the smaller one because it's only part of that. It's only the protons. So this 0 is the charge, but not the weight. These have heft. The reason we don't count the electrons in there is because the electrons are so minuscule, tiny. They're such a fraction of the size of protons or neutrons that their contribution to the weight is negligible. So they have a contribution to the mass of 0. So it would really be like 0.00000000001. Lots of zeros before we got to that 1. They're that much smaller, but they're really important as you learn when you learn about bonding. So knowing the atomic number, the atomic mass, you should be able to figure out the number of protons, electrons, and neutrons. We don't spend a lot of time talking about isotopes, but what you should know are that isotopes are variations of the same element. So the way we define what an element is by the number of protons. So as long as I have the same number of protons, doesn't matter if I have more or less electrons and become an ion, or more or less neutrons and I become an isotope. If I have the same number of protons, I am that element. So if I have 6 protons, I am always going to be carbon. So what an isotope is same number of protons, but a different number of neutrons. So the number, the mass number that we're given is for the most common combination of protons and neutrons that we find. So isotopes are only a small percentage of the total. So there's carbon-14, but it's just a small percentage of all the carbon, the way that we normally find carbon in the environment, which is carbon-12. Radioactive isotopes are those that can give off some radiation, some particles, gamma or beta particles, which would be subatomic particles that can cause damage to living tissue, to cells, that can break or damage DNA and cause mutations. We can use these as tracers because they act chemically. Carbon is carbon, it still acts like carbon. So if I have one that's giving this radioactive, these particles, it's emitting these particles, and I use that in a substance, it'll work the same, it'll be incorporated into the thing, but it's giving off those particles that can be tracked. So we use those as trackers. We use those a lot in medicine. We can compare ratios. So it goes into that a little bit more, but there will probably be an example, since in your objectives, when we point to a specific page number, it means there's probably some questions in there that could come up on this. So make sure you take a look at the discussion in your book on page 23 of Fukushima, which is the nuclear power plant that was damaged during the tsunami in Japan a few years ago. Let's see. So you should be able to describe that Bohr model of the electron with the electrons circling in those orbitals, or also called electron shells. The valence shell is always the outermost shell. And our octet rule, so for our purposes, the beauty of organic chemistry is that we are only looking at, well, for our biological molecules that we're looking at, we're really looking at smaller molecules. So we really only need the first 3 shells or orbitals. So here's my nucleus. I've got 1 orbital, 2, 3. So we can talk about the octet rule, because those outer or valence shells are always full if they have 8 electrons. They can't carry any more. The inner one is special. That first shell can only carry 2. So hydrogen with 1 electron just has 1 orbital with 1 electron in it. Helium with 2 has 2 circling. And then from there, I get that second shell, which can carry up to 8 electrons. So the valence shell, the outer shell, the orbitals fill with the maximum number of electrons that they can carry, closest to the nucleus first, and then as we move away, they start to fill. So they're always going to be filled. I'm never going to be missing one on an inner orbital if there's one on the outer one. That would move to that inner space, because the positive charge in the nucleus attracts that negative charged electron. It's going to move closer if there's room. And then my elements can come together to form molecules. So I can have 2 of the same element come together. Oxygen always comes as dioxide, O2, in natural forms, and that's so that it can fill the octet rule. So elements form bonds, relationships with one another, so that they can fill those outer valence shells with 8 electrons. So molecules are combinations of elements. If we have an element like or a molecule like H2O or CO2, where I have 2 different elements, I could call that a compound, which is technically what it's referred to, but we don't say a water compound, we say a water molecule. So molecule can really be used interchangeably. The book talks about compounds. We can use either one interchangeably, but molecules is the more commonly used word for any of these. So if we look at a chemical formula, it tells us how many atoms we have in the molecule. So for example, and I'm going to use this specifically because this is the one compound or molecule that we'll keep coming back to a lot in biology, and so you should recognize this. This is glucose, C6H12O6. So by looking at this formula, I know it has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. Chemical equations. I always write chemical equations with an arrow. And so when I write, if I have an arrow that is pointing to the right, then in this chemical equation, the reactants are what go in. So they're on the left side of the arrow. The arrow points to what comes out of this reaction, what's produced by it. So these are the products. So the arrow always points to the products. Sometimes I can have reactions that go either way. So if this was my reaction, then those would be the products. And these would be the reactants. So we always want to go what the arrow points to are the products. Typically, those are written the way that I had it initially, with the arrow pointing to the right and the products on the, or with the reactants on the left and the products on the right. The arrow points to the right. Oh, so ions. We looked at isotopes, which have the same proton number, but a different number of neutrons. So they have the same electron number as protons still, they just have different neutrons, so their mass differs. Ions are the same element, but they are either missing or have an extra electron. And so when we have ions, we would designate this, for example, if hydrogen was missing one electron, if it was a hydrogen ion, we would write it like this. It's hydrogen, but it's a plus. If you see a plus or a minus superscript, it tells me that, oh, this has a charge. Normally, there's no charge in my element. I'm neutral because I have the same number of electrons as protons. So I have one electron, one proton. Here, a hydrogen ion means I've lost an electron. So I have one more proton, one more positively charged proton than electron. I could also have a negatively charged ion. If I have a negatively charged ion, this is a chloride ion, so chlorine would have the same number of protons and electrons to have a neutral charge. Chlorine can gain an electron, which means it would have one more negative charge than positive. So it gains the electron, it gets more subatomic particles, but it has a negative charge. So this becomes a negative ion. So that positive or negative tells me which one I have more of. If I have more protons, it's positive. I have more electrons, it's a negative ion. So a positive ion and a negative ion. Let's see, compare polar covalent. All right, so our covalent bonds, our ionic bonds, oh, that's what this is in this one. Ionic bond is because if I have, for example, sodium as a positive ion and chloride as a negative ion, they want to be neutral. The positive, those charges attract like a magnet. Positives, opposites attract, and that the opposite charges pull them together so that when they're close together, they become neutral. Let me draw this in here. They become neutral because those positives and negatives cancel each other out when they're close together. So an ionic bond is really a fairly loose bond because it's just that attraction draws those two ions close together. So they're not really sharing anything, they're just drawn together because opposites are attracting. And the way I would form that, how I look and tell if I have that, is if the outer orbital is just missing one electron. So I always have two different elements involved in ionic bonding and only two. So I'm never going to have glucose C6H12O6, three different elements making an ionic bond. It's always going to be 1 to 1. And one of those is always going to have the same number of available spaces in its outer orbital as the other one has electrons in its outer orbital. So if my sodium only has 1 electron in its outer orbital, my chloride has 7. This one needs 1 to meet the octet rule and have 8. This one has that 1 lone electron. So in order to have 8 in its outer orbital, it would have to find 7 new electrons, or it could just kick that one out. And now its outer orbital moves down 1. It's only got 2 now, but that second one, becomes its outer orbital and its octet rule is filled. So one is going to steal or take away the electron from the other so that both satisfy the octet rule, and then their opposite charge draws them together. That's different from covalent bonds. So ionic bonds are the easier one to figure out. So you can always just look at the number of electrons, draw the elements, see if they have that corresponding relationship of 1 to 1. And if not, you're good to guess that it's going to be a covalent bond. And covalent bonds mean I'm sharing my electrons evenly. So in order to have full outer shells, these electrons are going to spend part of their time around one molecule and part around the other. And waters are a really good example of polar covalent. And our water molecule, we say we have a covalent bond because my oxygen has 6 electrons in its outer shell. It has 8 electrons total. Its atomic number is 8, so 8 protons, 8 electrons. 2 of those are in the inner orbital, so that makes that full. So it has 6 in the outer orbital. So I have 6 electrons in that outer orbital around my oxygen. Each hydrogen only has 1 electron. So the atomic number is 1. There are no neutrons in hydrogen. So I have a proton in the nucleus and an electron outside. So to fill that first orbital, I need 2 electrons. So my hydrogens are looking for another electron. And so they are going to share 2 hydrogens will come into that oxygen. The oxygen has 6. It needs 2 more. It's going to take 1 from one hydrogen, 1 from the other, but it's not going to steal it away. It's going to share. So those electrons are going to spend part of their time around oxygen and part around hydrogen. And then some of those oxygen, 2 of those oxygen electrons will spend part of their time around oxygen and part around hydrogen. So they're kind of zigzagging around. They're looping around, doing figure 8s. And that kind of draws them together. And because they're sharing those electrons, I think of the electrons like a needle and thread in this case. They're tying those together. So this is a stronger bond. And it's polar because the oxygen's much bigger. And so those electrons spend a lot more time around the oxygen than the hydrogens. And so those electrons are negative. They spend more time on the oxygen. So the oxygen has a slight negative charge on one end and a slight positive charge on the other. So we say it's polar. I have poles, a positive pole and a negative pole. Nonpolar covalent is I'm sharing all those electrons evenly. So nonpolar covalent, I don't have any positive or negative pull on any side of the molecule. It's even everywhere. Hydrogen bonds are formed when I have polar covalent bonds, or when I have any kind of polar molecules with a slightly positive charge on one side and a slightly negative. So if my hydrogens have a slightly positive charge, and the other side of the molecule, whatever it is, has a slight negative charge, I'm going to have hydrogen bonds form between the hydrogen, the slightly positive hydrogen, slightly negative other part. Hydrogen bonds, so my polar covalent, my covalent, my ions, this is how I form molecules. Hydrogen bonds are going to connect different molecules. So here's one molecule of water, here's another molecule. I'll get this loose connection of a hydrogen bond between the positive hydrogens of this water and the negative oxygens of this. So these are easy to break bonds. It's just a slight attraction based on that hydrogen slight charge. So this has to do with having that slight charge on my hydrogen atom, and it's going to connect molecules. That's how I get surface tension in water. Let's see, the five main properties of water because of hydrogen bonding. Everybody did a good job describing these in the discussions. So my water has high heat capacity. I have to put a whole lot of energy in order to raise the temperature of water just 1 degree. So when we call that a calorie. To raise 1 gram of water 1 degree, I have to put in a lot of energy, one calorie. High heat of vaporization is it takes a crazy amount of energy to get water to change form from a solid to a liquid or from a liquid to a gas. So it takes 540 calories to evaporate to make 1 gram of water turn from liquid to steam. That's why perspiration is such a good coolant, because for every gram of moisture of perspiration that evaporates off of me, it draws out 540 calories of heat. And that's why you say, oh, dry heat, it's so much better. It's dry heat than moist heat, because we can't cool off using evaporative cooling in high humidity. It has to be dry air for the water to vaporize. Universal solvent, water can dissolve more substances than anything else. And this is solutes. We're going to talk about solution solutes, solvents. I'm going to talk about that a little bit more in a subsequent chapter. So we will talk about this some more. And that fifth quality is that water is less dense. Oh, I jumped over adhesion and cohesion. Adhesion is that water sticks to other molecules, other types of molecules, and cohesion is that water molecules stick to each other. That's what gives us our surface tension on water. Somebody used the example of skipping stones. That surface tension is what lets us skip stones on water. Adhesion, that water running up the side of the graduated cylinder, that's from adhesion, the water sticking to the glass column. So we have adhesion and cohesion. And then our water is less dense as a solid than a liquid. The last portion of this chapter had to do with acids and bases. So we're going to remember the scale. High acidity means a low pH number. That trips people up. If it's highly acidic, it means I have a low pH number. So numbers below 7 are more acidic, because my pH scale is a measure of hydrogen ions. It's hydrogen potential. So the more hydrogen ions I have, the more acidic I am. But it's the inverse number. It's the fraction. It's 1 over how many hydrogen ions I have. So the more hydrogen ions I have, the smaller this total pH number is. That's why the low numbers are high acidity on the pH scale. And then bases are the high numbers. So numbers higher than 7, and 7 is neutral. Acid rain, make sure you read the information on acid rain, and look at the graph on page 32. So acid rain is defined as rain with a pH of less than 5. And we get that from carbon dioxide in the atmosphere. So the rain picks that up. It lowers the pH by giving us more hydroxide or binding with the hydroxide ions. So we decrease, so it ties up these, making this relatively higher, so giving us acidity, a lower pH. And some of the impacts that are talked about in there is that low pH, acidity, it can end up methylating mercury in aquatic systems, and that's a toxin that can bioaccumulate, so that it can accumulate into the humans through the food web if we eat things that it's accumulated in, and it is a neurotoxin. When the soil pH is low. If we have acid rain falling, that falls on the soil and lowers its pH, makes it difficult for plants to grow. Acid rain falling on leaves damages the leaves. It hinders their ability to photosynthesize, so they can't gain energy, and without that energy, they can't grow. Acid rain can also increase instances of asthma in humans and cause damage to structures, to buildings, to cars. If you see cars being shipped on trains or on car carriers and they have that white plastic on the roof and on the hood and the trunk, that's actually, they started doing that when acid rain started to become a big issue in the 80s. It's to protect the paint. And then buffers. So in your lab, you looked at acids, bases, and buffers. Buffers prevent a pH change. They do not try to make a solution neutral. They prevent a pH change. So if I add a buffer to a solution that is pH 3, and then I add acid to it, the pH should stay 3 because a buffer prevents a pH change. If I add a buffer to a pH 10 solution and then add acid, the pH should stay 10 because it buffers or prevents a change in pH. If I add more base to a pH 10 solution with a buffer, the pH should stay 10 because it's buffering, it's preventing any change to pH. Questions on chapter 2?
Nope.
Okay, good. That's usually the one that people worry about. Here, I think the lab really helped a lot of people for chapter 3 with the organic biological macromolecules. And in the discussion, everybody was able to define organic. When we call these molecules organic, are carbs, lipids, proteins, nucleic acids. The reason they're defined as organic, what we study in organic chemistry, are molecules that have lots of carbon-hydrogen bonds. So depending on what they are, they might have other elements. So if they're carbs, they're going to have carbohydrates, hydrogen and oxygen. If they're lipids, they're going to have carbon, hydrogen, oxygen. If they're protein, they're going to have carbon, hydrogen, oxygen, nitrogen. And depending on what amino acids, they probably have some sulfur and phosphorus. Some lipids are going to have a phosphorus with them. Nucleic acids are going to have carbon, hydrogen, oxygen, phosphates, and nitrogen. So there are different combinations to make these. Most of these have oxygens with them, but not all organic molecules do. They are derived from living tissue, from living materials. So schnapps, but as they break down, they lose the oxygen fairly quickly. So petroleum hydrocarbons are just long chains of carbon and hydrogen, or rings of carbon and hydrogen. So we don't have to have oxygen present. That was a common mistake in the discussion. So inorganic can contain carbon, like carbon dioxide, but there's no hydrogen attached to it. And then the 4 main categories of biomolecules are carbohydrates, the monomers are monosaccharides, the polymers are polysaccharides, our lipids. Our monomers typically are glycerol and fatty acids. This is a little wonky group because sometimes we have that ring structure with sterols. Waxes are long fatty acids. But our 2 big groups for biology, well, for our purposes, are the triglycerides that are used for energy, in fats and oils, and the phospholipids. The phospholipids are the main component of our plasma membranes and cells. So those have glycerols with fatty acid tails. And that's our main building block, and we just put those together. Proteins, the monomers are amino acids. I put those together into long chains called polypeptides. They're called that poly because there's many peptide bonds. That's what holds the amino acids together in that chain. It's not a protein. They're in the category protein, but the protein has to have a very specific shape. So until the polypeptide folds to its correct shape, the polymer is just a polypeptide. Nucleic acids are monomer. The building block is nucleotides. And those are made of a 5-carbon sugar with a phosphate and a nitrogen-containing base. So that's my main building block. I stack them together to get nucleic acids like DNA or RNA. Let's see. Dehydration, this always throws people because dehydration sounds like we're taking something away. So people want to think that that makes it smaller, but we take away a water to make a bigger molecule. So we remove a hydrogen from one of the monomers, a hydroxide from the other, so 2 H's and an O. We dehydrate or condense by removing that water, and that joins the 2 monomers together. So this is how we make bigger molecules, is through dehydration synthesis, or a dehydration reaction, or a condensation reaction. You can use any of those names. And we break down our polymers, our big long chains of repeating units, by adding a water. So lysis, it means to break down. So we're going to break it down with water. I add a water and that splits it apart. Let's see. So for carbohydrates, everybody should know the basic formula is 1 carbon to 2 hydrogens to 1 oxygen. And our carbohydrates have between 3 and 7 carbons, so between 6 and 14 hydrogens, and then 3 to 7 oxygens. There are a few exceptions. Deoxyribose, when we talk about DNA, the name tells you it's ribose. It's similar to ribose, a 5-carbon sugar. But ribose is a 5-carbon sugar, so they should both be C5H10C5. And it's okay, we have some with the same chemical formula, but when we look at their structure, those carbons would be slightly different, or where their hydrogens and oxygens are attached would be slightly different. Deoxyribose has 5 carbons, 10 hydrogens, but only 4 oxygens. Deoxy, it's down 1 oxygen. But for the others, they typically follow that 1 carbon to 2 hydrogens to 1 oxygen. You just know disaccharide. So when you see saccharide is sugar. So di, 2 simple sugars, mono, 1 simple sugar. When I have a polysaccharide, I put all those sugars together. Once I get a big molecule like that, it changes its chemical composition. So this is no longer considered a sugar, but a polysaccharide or a complex carbohydrate. So the examples of those are starch, glycogen, cellulose, chitin, peptidoglycan. These are all big molecules with repeating sugar units. Our lipid groups are fats and oils, which are the triglycerides. Saturated versus unsaturated fats have to do with whether or not they're going to be liquid or solid at room temperature. Glycerol is the component of the fats that is hydrophilic. It's attracted to water. The fatty acid tail portions are hydrophobic. They do not like water. They want to get away from the water. That's why fats will curl up. They get those tails away from the water, and they won't mix with water. And then our steroids, which are 4-ring structure, and waxes. We don't spend a whole lot of time talking about these. Our waxes are used for waterproofing, and steroids, our sterols, can be chemical messengers. Cholesterol is the big one that's found in the membrane of animals, and that's job is to keep the consistency of our membrane the same regardless of temperature changes. Proteins. Proteins do lots and lots and lots of things in our body. Here's the big one right here. This is the one everybody should remember. We're going to spend lots of time on this. Metabolism. The role proteins play in metabolism is they are enzymes. And enzymes are proteins that speed up chemical reactions. Remember, the definition of metabolism is the sum total of all the chemical reactions in a cell or in a body. And that means building big molecules from smaller ones and breaking down big molecules into smaller ones. So it's both sides of that. I eat food, I have lots of big molecules, I break them down to smaller molecules, and then my body uses those smaller pieces and builds whatever big pieces it needs. All those chemical reactions would happen anyway, but not at a fast enough rate to sustain life. So enzymes speed up chemical reactions up to 1,000,000 times faster than they would occur anyway. And that's necessary so that I have what I need when I need it to sustain life. So without our enzymes, nothing would happen. These are the big ones. They can be structural support, so strong bones and teeth, keratin, our hair, our nails. They can transport molecules. We have proteins in our plasma membrane that allow transport. They act like doorways to move things in and out of the cell. They're used for defense. Our antibodies or immunoglobulins are proteins. Some hormones, I'd mentioned hormones up here, the sterols, but we also have some hormones that are proteins. And then movement. So my muscles move because actin filaments and myosin filaments allow those cells to contract and relax and move past each other. So my cytoskeleton, moving things around inside my cells that we're going to learn about, are proteins. So those are the 6 roles of proteins. And you should be familiar with the 4 different levels of shape, how we get from a polypeptide to a protein. So my chain of amino acids, this is my primary structure. I have amino acids all in a row. So I have there a chain of amino acids. And then my secondary structure is because of attraction of the different electrons in the different molecule or elements that make up the amino acids. I'm going to either twist around and become a helix, or I'm going to kind of bend into this pleated shape. So I have a secondary structure that all depends on the order of amino acids. And then I'm going to fold in on myself to get a very specific tertiary or 3-dimensional shape. That's my tertiary structure. And it's very specific to what that protein's job is going to be. And then quaternary structure would be if I had multiples of these that come together. So hemoglobin has 4 tertiary structures that all come together to make one hemoglobin molecule. Not all proteins have that 4th level, but very large ones do. That shape, their final shape in their final configuration, now they're a protein. They're ready to do their job. And that shape is very, very specific to the job they do. Without having that shape, they can't do their job. So for example, if this is a protein and its job is to carry this, it's the exact right shape to do that. If one of those amino acids in the primary structure is out of order, it's going to have the wrong shape. And now it can't carry this molecule because it's not the right shape. So that those 4 different levels of structure, you should be familiar with those. The last group we talked about in my lecture video, just the basic structure, the nucleotides, that's my small building block, my brick. And I stack that up in different configurations. If I make 2 long stacks in opposite directions, I get DNA. If I make a single stack, I get RNA. And we're going to talk a lot more about those coming up when we get to our genetics section, when we start talking about DNA in a coming up unit, in unit 4. And then ATP we'll be talking about in our next unit on energy. ATP is a nucleotide. So it's a nucleic acid, but it's just a single nucleotide and it's modified. So it's got adenosine. So it's got ribose. Adenine is the nitrogenous base, and then it's adenosine triphosphate. So 3 phosphates. That's the modification. Usually they only have a single phosphate on each nucleotide. And ADP is adenosine diphosphate. That's when I only have 2 phosphates, and I have that because one of those will break off to release energy for the cell. Questions on chapter 3?
No, ma'am.
All right, then let's move on. So chapter 4, there's a lot of information in here. Most of this, everybody should have talked about cells a little bit in high school biology at least. So at least some of the words should be familiar. Probably eukaryotic cells because that's what we are. A little bit more than bacterial cells, prokaryotic cells. So this prefix means early or before, and eu means true. And karyote, this means the nut or the seed. So if you look under a microscope, that nucleus looks like if I cut a peach in half and it had the seed in the middle. So that's where that name comes from. If I look under the microscope, I see that seed in the middle. That's the nucleus. This is early or before there was a seed, before there was a nucleus. So these are my simpler, smaller structures. So you know that all cells, prokaryotes and eukaryotes, some of the things they have in common, what all cells have: cytoplasm, a plasma membrane, or cell membrane, same thing. They all have DNA, and they all have ribosomes. So whether you're prokaryote or eukaryote, you have those things. And then there are different prokaryotes have some different specific structures that not all eukaryotes have. All prokaryotes have a cell wall. Only plants and fungus have a cell wall in eukaryotes. So knowing those differences, eukaryotes have membrane-bound compartments. They have a membrane-bound nucleus. Don't have that in a prokaryote. These are the simpler ones. So you should know the 3 basic principles of the cell theory. So the 3 basic principles are all organisms are composed of cells. If we don't have cellular structure, we're not considered a living organism. We have cellular structure. That's why viruses aren't technically considered living. They don't have all the components of a cell. Cells are the basic unit of structure and function of all organisms. Everything that we do in our body is because each one of our cells is doing its job. So that's what that means. And then cells come only from pre-existing cells. This means life does not arise from non-living things. That's called abiogenesis, or spontaneous generation, that living things just spontaneously pop up. You got to come from a pre-existing cell. When we look at since these are on the objectives, you should probably know. Schleiden and Schwann, these were 2 German researchers. They worked at about the same time in separate labs, but they did compare notes and got together. Schleiden first observes that plants are composed of cells. Schwann kind of had that same discovery. He said, hey, look, all animals are composed of cells. Then they got together and said, hey, all living organisms are composed of cells. Our book talks about Virchow, who published a paper saying all cells come from pre-existing cells. Our book misses out. Remak actually published that 3 years earlier. Virchow plagiarized him. He gets all the credit. But at the time that this happened, the mid-1800s in Europe, there was a lot of anti-Semitism, and Remak was Jewish. And so he did not get credit for that until much later. So Virchow remains in the textbooks because he did publish it and get credit, but now it's recognized that Remak actually published it first and should be who that statement's credited to. Just little aside there. That's not in our text, but all right, let's see. Then you should know some prokaryotic structures. All cells have that plasma membrane. Bacteria always have a cell wall. The glycocalyx or capsule, these are similar names for the same, or different names for the same structure. It's like a sticky coating that some bacteria produce to protect them. Cytoplasm, all cells have that. All cells have DNA. Prokaryotes do not have a nucleus. But that DNA is in a ring that kind of crumples up, and wherever we find it is more dense if we're looking under a powerful microscope. And so we say they have a nucleoid region. So that region can float around in the cytoplasm any old place. Wherever we see it is the nucleoid. It's the region where the DNA is. And even though all of their chromosomes, all their genes are on that one single round chromosome, they can have extra chromosomal pieces of DNA. These are called plasmids. All cells have ribosomes, cytoprotein synthesis. Bacteria can have flagella. They're physically, structurally different from eukaryotic flagella, but same purpose. We use them for movement. And then fimbriae, some prokaryotes have these little thread-like structures. They're not cilia. They're not the same thing as cilia. They're not used for motility, right? Our cilia and our eukaryotes beat like oars. The fimbriae are just threads on the outside. And what they do is they let the bacteria grab on and get tangled up and stick to things. And then the conjugation pili is a modified fimbriae that's used for bacteria to be able to exchange bits of DNA. So usually they exchange these plasmids with one another. Conjugation's a little misnomer. This is not related to reproduction or cell replication or binary fission, how prokaryotes divide. So there are no male and female bacteria, and there's no sexual reproduction. So this is a little bit of a misnomer. They just share pieces of their DNA with this. Endosymbiotic theory, you'll want to take a look at that. This is our mitochondria and our chloroplasts. Probably through evolutionary history, it's been demonstrated that those had been prokaryotes. They've now given some of their DNA to the nuclear DNA. They've had this partnership. We've learned about symbiotic relationships. We've had this partnership. Eukaryotic cells have had this partnership so long, and it's become so close that now these formerly what had been bacteria have become part of eukaryotic cells. But they divide separately. They divide by themselves prior to cell division. They have their own DNA, which is in a single circle like bacterial DNA, and they have bacterial ribosomes. So they really technically have all the structures of bacteria, and that's how we were able to deduce that when we had good enough technology to identify these as bacterial, to see that this is how they started off. And then that partnership, that symbiosis, became a permanent partnership. They became an organelle. The chloroplasts and the mitochondria became an organelle inside a larger cell. When we get to our eukaryotic cells, nucleus is a membrane that houses the DNA. Chromatin is the material of our chromosomes. So it's DNA plus proteins. Chromosomes, that's our DNA. And we have 46 chromosomes. This just is the word that refers to how our chromatin is packaged as. Our genes, it's the information, an area along a chromosome that carries our trait information. The nucleolus, this one kind of breaks brains a little bit if you think about it. The nucleus is a region inside, the nucleolus is a region inside the nucleus that when we look under the microscope, it's darker because it's more dense, because it's filled in with lots and lots and lots of molecules. It's filled with RNA, ribosomal RNA, and proteins. And that ribosomal RNA is going to go out from the nuclear envelope into the cytoplasm through the nuclear pores, and it's going to form the ribosomes. So this is where the subunits of the ribosomes are built, inside the nucleus. And then the nuclear envelope, that's my membrane that encloses my chromosomes. And then you should also know these structures. I think the lab does a great job. Everybody should be on or finishing up. I think it's lab 5 that's on cells. So that's a really great way to learn all of these different portions of eukaryotic cell. vesicles, are little membrane-bound transport units. The cytoskeleton moves things, proteins that move things around the cell. Ribosomes, the site of protein synthesis. My endoplasmic reticulum is an extension of the nuclear envelope, so membrane-bound organelle, and it is the site of lipid synthesis and protein synthesis. The rough ER has ribosomes on the outside, so that should tell you, oh, that's where proteins are made. And the rough ER does not have those, or the smooth ER does not have those ribosomes. That's where the lipids are made. And then that membrane-bound organelle can break off little vesicles so it can surround the lipid that it made or the protein that it made in a membrane. Just like you can break off little smaller bubbles when you blow bubbles. So these are like bubbles that break off from the membrane and transport those proteins and lipids to the Golgi apparatus, which is a membrane-bound organelle that's going to package and process those proteins and lipids into complex molecules, into their final shape. And then it's going to break off those transport vesicles to ship those molecules to the cell membrane or then out of the cell to expel them out of the cell. And if you didn't watch my lecture video on that, I walk through an example of how that process works with the endomembrane system. Lysosomes and peroxisomes, these are garbage disposals. So lysosomes contain digestive enzymes. So when the cell has to get rid of molecules, it ingests something bad or needs to get rid of a bacteria, a viral particle or something that infects it, or it makes a mistake when it's building a protein or a lipid, this is the garbage disposal unit. It will use that lysosome to break down the garbage to its building block pieces and then eject it from the cell. Peroxisomes are garbage disposals specifically for toxic oxygen products. So vacuoles are little storage membrane-bound storage units. The central vacuole is a specific storage unit found in plants that dominates what we see in a plant cell. It's a big bag that is used for water control, for controlling water in and out of the cell, and also waste products, because some large waste products can't get out of plant cells because we have a cell wall. It's a wall that prevents things from moving in and out. And so some waste products are also stored in that as well as water. These are my actin filaments, intermediate filaments, microtubules are all of my different proteins that are involved in moving things around the cell. They're parts of the cytoskeleton. Centrioles are my microtubule coordinating center, making sure these go and move things to where they need to move things. And then cilia, small, short, used like oars for movement, and flagella. In eukaryotes, the flagella has a different structure than in prokaryotes. In eukaryotes, the flagella moves like a whip, and that's how it moves the cell. The flagella in prokaryotes and bacteria works like a rotor or like a propeller. It spins. And then the last organelles that we looked at in this chapter were the chloroplasts. We're going to look more closely at those in the next unit when we take a look at photosynthesis and cellular respiration. So the chloroplasts are found in plants, and they are the site of photosynthesis, where I build glucose molecules for my plant. So this is plants, autotrophs, self-feeders. They feed themselves glucose that they make through photosynthesis. And the mitochondria is the site of energy production in the cell. Here's where I take that glucose and I break it down to get ATP so the cell can use that energy to do work. Animals get their glucose from the food they eat. and then they send it to the mitochondria to be broken down. Plants make their glucose through photosynthesis and then send it to the mitochondria. So really, really important you remember, plants have both of these. It's not plants have chloroplasts, animals have mitochondria. Plants have both of them because all cells need a way to get energy. So mitochondria are found in plants and animals and fungus and protists. Chloroplasts are plants only. All right, question about any of the cellular structures?
Nope.
All right. And then our last chapter is chapter 5 on membrane transport. So our plasma membrane, this is what separates the inside of the cell from the outside. For anything to get into the cell or leave the cell, it has to cross this membrane. And it's a phospholipid bilayer. So we have 2 layers of phospholipids. Remember that glycerol head with 2 fatty acid tails? And it's a bilayer, so we'd have one whole layer of these and then another layer of these. Those fatty acid tails are hydrophobic, so they want to get away from the water. So they turn in so they're not touching the water. The glycerol heads that are hydrophilic are touching the water. And that water is the cytoplasm, because that's aqueous solution, or the external fluid around the cell, which would be an aqueous solution. So those tails turn in. So that's why we have a bilayer. And then we have proteins floating within that. So that gives us our fluid mosaic model. The mosaic is a picture of lots of different tiles. This is lots of different tiles. We have the background would be all those phospholipids, and then those proteins would be the other tiles in there that make the picture. So it's a fluid mosaic. It's the consistency of olive oil, so it really is fluid. Those things can move and float around. So phospholipids are the main component. A little bit cholesterol is the sterol. Its job is with temperature fluctuations. If you think about that's like it's a consistency of olive oil. Well, if I got really cold, olive oil kind of gets really viscous and thick and doesn't flow so well. Or if it gets really hot, it gets really runny. Cholesterol makes sure that we have the same consistency of that membrane across all temperatures. Whatever happens, it's going to stay that same consistency. We're going to talk about membrane proteins here in number 3, but those are embedded throughout this the membrane and the phospholipids. And then my glycolipids and glycoproteins. That lipids, these are those phospholipids. Some of them have a sugar attached. The proteins, part of that fluid mosaic, some have a sugar attached. The branching, the type of sugar, the size of the sugar, these molecules that are sort of attached to the lipids, to the phospholipids, and to the proteins, to some of them, are very, very specific. Their shape and structure is unique to each individual. So it really gives us cellular identification or recognition. That's how our immune system knows. When our immune system is monitoring for threats, it knows, oh, this cell belongs. Oh, this cell belongs. This cell belongs. I'm not going to attack you because you're part of me. That's how our immune system cells recognize us. This gives our cell a fingerprint. So that's when you hear about how transplant patients reject a transplant. We have to have the right type of blood transfusion. It's because we have to have that right fingerprint on our cell surface. So that's what the glycolipids and glycoproteins do. So those proteins in my fluid mosaic model, they have 6 different jobs. Some of them work as channel proteins. And what a channel protein is, it serves as a doorway. So some molecules can just cross through the phospholipids in and out, but they have to be very small nonpolar molecules. If they're too big and they can't pass through, then I'm going to have to give them a doorway. Channel protein's a doorway. If you can fit, you can move across that. Carrier proteins are specific to specific molecules, so they'll only allow certain molecules to come through that doorway. Then cell recognition proteins, these would be a specific glycoprotein. So they recognize cells that do belong. Receptor proteins, these are on the cell surface, and they let specific molecules attach. They're the exact right shape that specific molecules can attach to them. So an example would be for hormones. Hormones are produced in one part of your body. They travel through your bloodstream. They don't have any impact on all the other cells they pass until they find one with the right receptor protein on its surface. And then they can connect. And then that specific cell is affected by that hormone. And then lots and lots and lots of the proteins on our membrane are enzymes. So enzymes, all the chemical reactions of a cell, lots of those reactions take place right at the surface because that's the interface with things coming in and going out of the cell. So that's the fastest place to let that reaction take place. See, selectively permeable, only certain things are allowed in and out. And the way things move in and out, they're going to move without energy down a concentration gradient, or they have to move up the concentration gradient, then we need energy involved. So I think a gradient is a slope. So if you think about it as a hill, if I go downhill, I don't need any energy. So if I go from high concentration to low, I don't need any energy. If I go uphill, I think I'm going to have to work. So I have to put energy in to go from low concentration to high. And then bulk transport, this is going to use energy because I'm moving, but I'm going to use that plasma membrane and that cytoskeleton to engulf. So if here's my membrane and I want these molecules here, I can wrap around them and pull them in. Or if I have one of those lysosomes with garbage ready to go out, and here's my membrane, it's going to move to the membrane, merge with the membrane, and then push things out of the cells. That's bulk transport. Endocytosis, bringing things in, or exocytosis, moving things out of the cell. So this chapter also talks about solutions, because we're looking at water specifically moving in and out of the cell. So remember, a solution is made of a solute and a solvent. My example is Kool-Aid. It's made of colored sugar. That's my solute that's dissolved in my universal solvent, water. When I give the concentration, if I'm talking about Kool-Aid, a more concentrated Kool-Aid solution would be sweeter. So the concentration refers to the percent of the solute, how much sugar is in there, not how much water is in there. And then we looked at diffusion and osmosis. I think everybody should have completed that lab on diffusion. Maybe you haven't completed it yet, but should be doing the diffusion and osmosis lab. Diffusion is simply molecules, any molecule moving from high concentration to low. It may be across a membrane. It doesn't have to be. It can just diffuse through the air, through the water. Osmosis is a very specific case of diffusion. It's the diffusion of water across a membrane. So the only molecule I'm concerned with is water, and I'm always looking at how it moves across a membrane. And then facilitated transport, this is also passive. So when I'm talking about passive, no energy because I'm going from high concentration to low, but here I'm going to use one of those membrane proteins. So I'm going to use a channel protein or a carrier protein to do this. Right, most important thing about hypotonic, hypertonic, isotonics. Isotonic, same concentration. This does not mean a 50% solution. So 50% solution is 50% water, 50% solute, so 50% sugar. That's not isotonic because these tonicity terms are relative terms. They compare two things. So I can't just talk about a solution. I'm comparing two different solutions. So 50% solute concentration, well, I don't know if that's hypertonic or hypotonic or isotonic. If I'm comparing it to another 50% solution, they are isotonic to each other. If I'm comparing that 50% solution to a 60% solution, then that 60% solution has more solutes. It is hypertonic compared to my 50% solution. If I'm comparing it to a 40% solution, and this is 50%, did I just say that? My 60% is hypertonic to 50. If I'm comparing it to 40, my 40% is hypotonic compared to the 50. If my 40% is hypotonic compared to the 50, that means my 50% solution is hypertonic compared to the 40. So if I have one of those words, hypertonic or hypotonic, then I also have the other because it's a comparison. Okay, so isotonic means they're the same to each other, but the other two, if one is hypotonic, lower concentration, then the other has to be hypertonic. So read carefully. This comes up in every class in the lab because they switch the wording on one of the questions. For some of the questions, they talk about a cell in a hypertonic solution. Well, if the solution outside is hypertonic, the cell must be hypotonic. But then they switch it on a question to give you a hypertonic cell. That means the solution outside of it must be hypotonic. So read carefully with those. And then the terms Turgor pressure. So Turgor pressure and plasmolysis refer to plants. If you think Turgor pressure, it's pushing up. When I water my plants, they absorb all that water. That central vacuole expands, pushes up against the cell wall, and my plant is all nice and rigid and firm. If I don't water that plant for a week, it's going to gradually start using up that water from inside the central vacuole, and that central vacuole is going to shrink. It's going to use the water from the cytoplasm, and my plasma membrane is going to pull away from the cell wall and shrink. The cell wall is going to keep its shape, but that plasma membrane and the central vacuole membrane are going to pull away from that cell wall, and that is called plasmolysis. This is a tricky term because lysis means to burst, but in this case, it's that plasma membrane is just shriveling in. And then in animal cells, because they don't have a cell wall, if they are placed in a hypotonic solution, water can keep rushing in until either equilibrium is reached or until the cell bursts. That's called cytolysis. So lysis, burst, cyto, the cell. In red blood cells, if we're looking at red blood cells, we call that hemolysis. If we put a cell into hypertonic solution, water is going to leave the cell. The plasma membrane is going to shrivel. The cell is going to shrink, and we call that crenation in animal cells. Let's see. Active transport, I need to put in energy because I'm moving across or moving up my concentration gradient. So sodium, potassium pump, if you see the word pump, that's going to be active transport. It needs ATP. If we look at bulk transport, we're moving that plasma membrane, that's going to require energy. So that's active transport. Exocytosis, I'm bringing things in. Endocytosis, I'm moving them out. When I'm moving in big molecules, call that phagocytosis. So phagophage, this prefix has to do with eating. So if I'm eating big molecules, phagocytosis. We give the term pinocytosis to taking in very small molecules or liquids. I remember this by associating that with Pinot Noir. If I'm bringing in wine, it's pinocytosis, so liquid and small molecules. And then receptor-mediated endocytosis, I mentioned those proteins that serve as receptors. Some of those, when the molecule that they're specific to lands there, they want to bring that molecule into the cell. So receptor-mediated endocytosis. That's actually how a lot of viruses get into cells. And then we have some cell surface modifications. And the big ones are, how do cells stick together? So my extracellular matrix, this is really connective tissue. This is stuff outside my cell. But then I want my cells to stick together. I have some different ways that my cells can stick together. Adhesion junctions, we're just going to make sure my cells stay together because I don't want my cells all falling apart. Tight junctions are a specific type of adhesion junction. So adhesion junctions typically are just connected in a few spots, those plasma membranes. So that gives me flexibility and keeps my cells together. So some of my organs, though, I want to make sure nothing leaks in or out of. Like, I don't want my stomach acid able to leak in or out. And those adhesion junctions, because of the flexibility, molecules can move in between the cells to get in and out. So my stomach, I don't want things moving in and out of there. My kidneys, I don't want things, there's certain, he says stomach is the big one with the acid, but where I really want to make sure that whatever is in that organ or in that tissue stays together, those we're going to have tight junctions. And then gap junctions are where I have those channel proteins lined up between adjacent cells so that I can have movement and communication among the cells. And then in plants, because they have a cell wall that would prevent anything, any kind of communication or movement between the cells. So they have little channels called plasmodesmata that are little openings between those cell walls so they can exchange materials. So that's some modification to that cell surface in plants. Any questions on this so far?
No, ma'am.
Okay, diffusion and osmosis tends to be the tricky one, but these labs do a really good job. But I know that always kind of catches people off guard when if you have one solution that's hypertonic, the other must be hypotonic. So you can change the wording to ask about what's on the other side of the membrane. So I can ask about the cell compared to what's outside, or I can ask about what's outside compared to the cell. So just make sure you read any questions really carefully that use these terms. All right?
Yeah, that tripped me up a little bit.
Yeah, that one usually gets quite a few people. It's like, oh, wait, they're asking, wait, they said in hypertonic solution that the cell would shrivel, but now they're saying, oh, the cell is hypertonic. So yeah, always just be careful to know which solution they're talking about in the question. I will, once this is processed, I will post it. So if you need to go back over anything, it'll be in the D2L classroom.
All right, thank you.
Thank you.
Bye.
Bye-bye. All right, stop sharing.